Tienes tu micrófono apagado.
Yeah, hi. Good afternoon, everyone, and welcome to the UNAM UK and MexSocUK student seminar series. My name is Farid, and I'm president of the Society of Mexican Students in the United Kingdom, and I'll be moderating today's session. We're delighted to have you join us for another session highlighting the work of Mexican researchers contributing to science and innovation from institutions across the United Kingdom. These seminars aim to foster dialogue, collaboration, and visibility of outstanding research carried out by Mexican scholars abroad. Today's speaker is Aranza Jhosadara Espinosa Flores. She is a PhD candidate at Queen Mary University of London, and she will present the talk titled Integrating CRISPR Cas9 and AI in Zebrafish to Accelerate Green Drug Discovery for neurobehavioral targets. Aranza holds a BSc in nutrition from the Instituto Politécnico Nacional, 6 Unidad Milpa Alta, and a master in science in health sciences from the Escuela Superior de Medicina at the same institution. Her early research explored the metabolic and gastrointestinal effects of artificial sweeteners, as well as some multidisciplinary treatment approaches for patients with neurological conditions and swallowing disorders. Currently, her doctoral research focuses on neuroendocrine modulators for appetite, combining genetic modification and machine learning to identify novel hemp-derived compounds with potential neuroactive effects. In today's talk, she will discuss how integrating CRISPR-Cas9 and artificial intelligence can accelerate green, sustainable drug discovery, using these zebrafish as a model organism for metabolic and neurobehavioral studies. Please do remember that you can leave all your questions in the YouTube chat throughout the session. They will be addressed during the Q&A at the end of the presentation. Please, Aranza, thank you very much for joining us, and the floor is yours.
Hello, Farid. Thank you so much for the introduction. Let me share my screen. Sure. Okay, can you see my screen? Good. So, yeah, thank you for the introduction. It's an honor for me to be part of this seminar from UNAM UK. I'm a PhD candidate and I'm studying in Queen Mary University of London. So first and before starting, I want to show a little bit of being grateful of the opportunity that I have here. And in the UK, specifically in London, I had opportunity to see all around UK. I also had the opportunity to bring part of my research to Belfast, Ireland. I have met many beautiful people, including my supervisor and colleagues that had helped me go forward into my research. I think if we do not create community alone, it's way too much more complicated being so far from Mexico. Also, I want to give a little bit shout out of Mexican woman, science woman. Here's a picture of Catalina and me. She is 1 year ahead from me. And when she was in 3rd year and I was in 2nd year, we both won the award of the best talk and the best poster. So yeah, it was a competition inside the school. However, I'm really proud that both Mexican female were highlighted into this context. And yeah, just this is just a collage of all my life in the last almost 3 years already. So yeah, just keep going. If you wanted to study in a foreign country, don't stop on your dreams and keep pushing until you get it. And of course, it's a compilation of have really hard work. So to start with, My topic is integrating CRISPR-Cas9 and artificial intelligence in zebrafish to accelerate green discovery for neurobiological targets. Basically, CRISPR-Cas, it's a technique, it's a well-known technique to modify genes. So I didn't put it in the presentation, but if you have a specific question about this technique, you can for sure ask me at the end of the session. So why AI and zebrafish help us discover green or sustainable neurodegenerative drugs? Here we have a picture of the anatomy of the brain of a mammal and a brain of a zebrafish. So they are different in terms of the anatomy, but they have conservated areas. We know that some specific areas of the brain have a specific function. So the same as works with mammals, in this case, they are a rodent brain. Here we have the zebrafish brain. Use of AI tools can significantly reduce the time and cost contributing to a more green, sustainable drug discovery process. A process that was back in the days, it took like 20 to 30 years to approve a drug because we need to test 100 drugs or 200 drugs in the lab and see which is the good one, and then proceed to test it in some complex organism, and then test it in a clinical perspective, and now in humans, and so on, all the phases of the clinical trials. So nowadays with machine learning or artificial intelligence help us to predict which of those 100, 200 molecules or possible drugs are the best, and we can just predict which is the best one and analyze it in the lab. Of course, we cannot forget about the lab completely. We still need to test those drugs and then proceed and make it way too much easier and faster. Part of the results that I'm going to show you are based on a project that I did in 4 months. So in 4 months, we did the artificial intelligence and then the testing of the possible candidates. Which are the possible candidates that I was focused on was the extracts of hemp. These are on underexplored complex chemistry. These are small molecules, specifically terpenes. We already know that hemp or cannabis sativa have different cannabinoids that can alter our brain. However, there are many other molecules in the hemp extracts that we have no knowledge about the possible effects in our brain. And last but not least, zebrafish is a scalable, fast vertebrate model with 70% sharing genome with humans. And as the picture showed, the central nervous system similarities to mammal for discovering neuroactive drug targets. So even though we think a fish, it's really small or it's so different from us, we share 70% of our genome. And even though there's this myth that zebrafish do not remember or have a lack of memory, in contrast, that's not true. Zebrafish can also, there are some papers that show that they can even count up to 3 or 4, I think. So it's a very interesting model to use and very cost effective. And so we're combining all these tools to make it easier and faster in the way of drug discovery. Sorry. Okay. So GABA as a receptor, as a possible hemp mechanism of action. So what is GABA? GABA is an ion channel binding, and this is the primary inhibitory transmitter. So this is GABA, just the signal in our brain that tells all the neurons to go slow. Let's say that is GABA. So GABA A, it's the receptor of GABA. And the principal target for the this is the principal target for the development of anxiety reduce drugs. So every time that, or most of the times, of most of the drugs that are focused on anxiety treatment, they binds with GABA. So the drug most of the times, or the one that I compare with, for example, is diazepam or any other benzodiazepines, they bind with GABA. That's what make the patients with anxiety to go, let's say, slow, to calm this anxiety symptoms. And GABA-A activity is influenced by various substances, including alcohol, neurosteroids, and cannabinoids. So there's already plenty of information that cannabinoids alter this GABA signaling. But as I already tell you, the hemp oil has other different molecules. And what is hemp oil? It's a variety of cannabis sativa that contains low levels of THC and high concentration of CBD, a non-psychoactive cannabinoid with therapeutic potential. And this often contains terpenes. These are aromatic compounds, not only in cannabis sativa, but in other fruits and vegetables. For example, the pinene is the molecule that makes the pines smell like pine. So this is what that put smell and flavor to different fruits or vegetables. And for example, in pinene has been described that pinene have a calming or anti-anxiety effect through the entourage effect, which is another term to use the binding effect of the CBD and other terpenes, or indeed the whole spectrum of the hemp oil. Unlike THC, hemp derivated CBD does not cause intoxication, and that makes it suitable for daily use and widely available. So how we can integrate machine learning and Zebrafish with GABA-A receptor? First, we needed to have a proof of principle. What did we do? So there are already databases that has all the information and all the molecules that binds with XY target. In this case, we use GABA-A. So we download all the data, all the molecules that binds with GABA-A that are already known from this database. Then we filter to discard the less, the pitch symbol, which is the pitch symbol is the affinity number. So less than 6, we discard them, and then we generate descriptors. So using RDKit package, this helps us to knowing there's smiles, which is, I call it the fingerprint of a molecule. So once you have the smiles, you put it into RDKit, and RDKit shows you or generate these more than 100 descriptors. including molecular weight, topological surface area, and so on. Many descriptors that describe the molecule in many different senses. Number of electrons, number of neutrons, etc., etc. So then we visualize it. Here we have a picture of it. We have the chembol with all the molecules that binds with GABA in blue, and the terpenes that are way farther from them. So with these, we didn't have like too many hopes, maybe a couple of them that are overlapping. You can see here and here. They are overlapping with the known molecules binding with GABA, but we keep working on this and try to make a prediction. And once that we take this this prediction from the machine learning, we tested in the fish. So how do we measure anxiety in fish? First, we cannot say that fish has anxiety because it's complicated to diagnose a fish, but as well as with other model organisms, we said that there's an anxiety-like behavior. So we have larvae of 5 days that are around 4-5 millimeters, and we put them into a 24 well plate. We have a beautiful machine that actually it's right next to me that can track the movement of the fish. So what we design is a template that involves the 24 arenas, so the 24 the arenas where the fish is gonna swim, and each arena is divided into 2 zones, the outer zone and the inner zone. So if the fish swims in the outer zone, that means that it's scared or it's anxious or it's showing an anxiety-like behavior, compared to the to when they swim in the center. If they swim in the center, they are not afraid and they are they are calm or less anxious. We also put them into a light on and off cycle, so they can also, they used to swim more in the light with the lights off, so we can see a difference in between how they move or how these change, which it's a stress for them, how this change of lights impact them. This would be like the stress stimuli. And so we measure two things: the time in the inner zone, let's say the time of that the fish swims in the center of the well, and we also measure the distance travel, how much the fish moves. Why? Because most of the anxiolytic or the benzodiazepine, in this case diazepam, show sedative effect. So if you're an anxious person, you need to have a very controlled dosage for you to still be productive, because they can be sedative, you can feel nauseous, and of course these are very create withdrawal, or in common terms, they become they induce an addiction of the drug or high dependence. Here we have just the diazepam. This is basically the proof of principle to verify that in our life, that in our lab we can test or we can replicate what other labs did before. So here we found that 10 micromolar, that's the concentration that increase the time in the inner zone. However, in the distance travel, from half of that amount, they show a reduce in the distance travel. So they didn't even have the anxiolytic effect, but they were having a sedative effect effect already. So that's the main effect, that's the main problem, that we want to find another kind of drugs that are not addictive and less sedative. So here we have a hemp oil extract, which involves CBD and many terpenes. And we can see here that it's actually the other way around in the terms of sedative or non-sedative. And from a very low dosage of this hemp oil, they start increasing the time in the inner zone. And yeah, you can increase the dosage, but from a very low dosage, they show less anxiety. And yeah, as I already mentioned, they contrary to benzodiazepines, they show an hyperactive effect. So as I mentioned in the beginning of the talk, we have alpha-pinene. This pinene is what contributes to the smell of pine, and it's already known that reduce anxiety. Also in zebrafish, a couple of papers mention it, and we also replicate, as always. We need to be sure that another study it's replicable. So we replicate it, and yes, they show an increment in the swimming in the inner zone from a very low dosage, as well as the hemp oil. And they, at that same dosage, they were hyperactive, and in a higher dosage do not contribute with anxiety and has a sedative effect as well. So we try three possible candidates coming from the machine learning prediction. And one was this beautiful terpene that due to it's confidential still, and I cannot say the specific molecule, but with this molecule, which is a terpene, we find out that the effect of the anxiolytic effect was 10 times low at a lower concentration. So the potency was 10 times bigger compared to the alpha-pinene, for example. And in the case of this, they didn't change this molecule didn't change the distance traveled. So does not has an hyperactive effect or a sedative effect at the dosage that is effective. It starts increasing to go hyperactive, but in a very high dosage. And once that we established that in GABA, I want to give like a little plot twist. I am focusing my research in MC4R. As Farid told you, I am a nutritionist, so it's a big of my interest, MC4R, because MC4R it's a G-coupled protein receptor in the hypothalamus that plays a critical role in controlling appetite and energy expenditure. And the mutations in MC4R gene, or when MC4R receptor do not work due to a mutation, it's the most common cause of monogenic obesity. What is monogenic obesity? Is that you just need to have an alteration of one gene to have obesity. There are polygenic obesity as well, when you need to have a specific 2 or more genes altered in your genome to altogether create obesity. But MC4R is just this. You just need a little modification of MC4R, and you can have obesity from a very early stage of life. Has also a role in maintaining glucose homeostasis and lipid metabolism. These are novel novel information that have found in rodents, and we were able to find it in the zebrafish as well. And yeah, so MC4R affects automatic functions such as heart rate, blood pressure, and modulates system sympathetic output. This is an in situ hybridization that I did in the zebrafish brain. You can see here that this is the brain, this is the cut, and this is how it looks like once you cut the brain. I stain it, and the purple areas is where the melanocortin 4 receptor is expressed. So part of my PhD was to start analyzing or going further into MC4R using this mutant line of MC4R. However, we find out that they didn't eat more. So this is the symptom number 1 of MC4R non-function, the increase of food intake. So we find out that this line that was supposed to be the one that I work with, they show no phenotype or no symptom expected. So we go into the genetic and the sequence of this receptor, and we have here, this is the sequence, and we use a beautiful paper that tells us what is the specific areas, or which are the specific amino acids that gives the property or gives the functionality of this receptor. So we find out that here, this stop is the mutation of the MC4R SA122. It's a stop, good, but there is a COSAC consensus sequence. What is a COSAC consensus sequence? It's different patterns of nucleotides that when they happens to be there, they can increase the possibility of a start codon. So if this is a 1 exon gene, so we genetics life, we always try to survive. So if there's a stop up, the machine that we are, it's gonna try to still work or make something functional, in this case the protein or the receptor. And yeah, we find out that the protein is still transcribing, and that's why we have no symptom, because the modification was not properly made, let's say. So we use CRISPR-Cas9. We inject the Cas9 with the guide. The guide is like the specific sequence of the gene. We tried, sorry to go back, we tried to make the guide around here that passes the COSAC sequence. So we inject, we have different mutated fish, we wait for them to grow, we outcross them with wild type, and we have some half mutated embryos. We predict different proteins, we find out which is the good candidate, and once I have the ones that my fish that have mutated, I in-cross them, because luckily I got 2 fish, 1 female, 1 male with the same mutation. So I in-cross them, and I finally got the line. This is just an image representation. What I do to measure how much do they eat, I put fluorescence in their food, and then I feed them with this fluorescent or dye label food, and that's what you can see in the microscope. You can see the belly full of food because they have eaten the fluorescent food. And once that I have the fish like this, I put it into a microplate reader and to have a quantitative data of the fluorescence of the fish. We can see here that that my line show an increment in food intake, which was very happy days. So we made the same in terms of the terpenes, but in this case, we don't use GABA, we use MC4R, and that's what we did. Same, all these molecules are the ones that binds with MC4R, and the red ones are the terpenes. So as well, there are a couple of matches, and we proceed to do the machine learning model evaluation. We made a tenfold cross-validation to know which would be the best model to use in this case. I use random forest because it has the lowest error, and yeah, that's how we choose random forest. And these are basically my results. Compared to GABA with MC4R, they do not show high affinity or not high affinity. Here is the graph of all the molecules, the pitch envelope or the affinity value, and how much the frequency of molecules binding. So we can see that most of the majority of the molecules have an affinity value of around 6.5, 7.5, let's say 7. And this is the distribution of the affinity value of the terpenes with MC4R, and some of them are small, let's say, they have a low affinity value. That doesn't mean that they are not affinity at all, because we can see here that there are some affinity, some binding binding molecules with lower affinity. So just show a low affinity values compared to the GABA ones. So summary and future directions. As a summary, with machine learning prediction applied in Zebrafish, we demonstrate a fast, sustainable method for identifying neuroactive terpene binding with GABA-A receptor for reducing anxiety-like behavior. We also know that SA122 mutant line is not a suitable model to study MC4R loss of function. We validate MC4R knockout model with a robust hyperphagia phenotype. We confirm anxiolytic effect of CBD hemp oil in zebrafish, and terpenes show a low affinity with MC4R. Which are the future directions is, well, we always have more questions. When you answer one question, you have another two to answer. So is hemp oil and terpenes effect due to MC4R? Those anxiolytic terpenes preserve their anxiolytic effect in MC4R knockout fish. Do we induce anxiety phenotype by overfeeding in MC4R knockout line? And metabolic and inflammatory findings in MC4R knockout fish. So I'm focusing in inflammatory. So right now that's what I'm doing, and I'm happy to share my results once I have them. And yeah, thank you again to my beautiful colleagues in the lab, my sponsors, and of course UNAM UK to open this opportunity to talk about this. And thank you.
Thank you very much, Jhosa, for this insightful and very forward-looking presentation. Well, with this work, it kind of reminds us that innovation could bloom from wherever where disciplines meet, genetics, AI, sustainable pharmacology. This is very multidisciplinary, as we mentioned earlier. Thank you very much again. And now we'll start with some questions. Firstly, I've got one. Um, so we, well, you might have mentioned it. I don't quite got it. So you've got zebrafish. This is quite a different animal overall from mammals. How do these models compare to some other mammalian models in predicting these neurobehavioral outcomes related to appetite regulations?
Um, of course, they are not as good as mammals because mammals are the closest the organism is to humans, the better. So that's why some studies they do it in primates or even in bunnies, in rabbits. There are studies in many model organisms. The good or the benefit of doing with zebrafish is that it's just one step under the mammals with very cost effective. It's way too much lower the price to maintain 20 fish, or let's say we can maintain 20 fish with the amount that we can maintain one rodent. And that increase our number of individuals that help us to, yeah, it's better to use 10 individuals instead of 3. And sometimes or most of the n numbers of rodents are like from 3 to 9 the most. So with the zebrafish we can test even 30 30 50 individuals. Of course we cannot use many because there's laws and everything. And in terms of neuro impulses or all behaviors, most of the if I could say all, but I haven't read about all, but most of the behavioral essays that are made in rodents, we can do it in zebrafish. This thigmotaxis, that's the color, that's the name of the essay of how the fish swim in the inner zone or the outer zone. That's also applied in rodents. But of course, with rodents, they need a bigger space, a bigger maintenance, rather than in zebrafish using a 24 well plates, I can analyze 24 individuals. And that make it so much easier. And in terms of CRISPR-Cas, in mammals, for being mammals, they need to extract the egg, extract the sperm, do the in vitro fertilization, and then put it back to the mom for the babies to grow. Because, yeah, the in vitro fertilization and the injection for the modification for sure, and then put it back to the mom, which decrease the survival rate and the time and all these procedures, rather than with fish that you just mate a female and a male, and the next day you can collect the embryos, and the ones that are in one cell stage, you inject them directly. So you don't need to put them back in the mom, you just rear those fish, and those fish, you know that they are genetically modified. Did I answer the question? Yeah.
Yes, you did. Yeah, you did quite well. Perhaps you might have answered some of my other questions, but that's fine.
Yeah. No.
So basically, we could use this as some sort of beginning of or the first models for this, and then if we make sure that everything's going according to plan, you could just step up to some very specific mammals, just that are closer to the human or to yeah.
Yeah, and also most of the embryology information that we know in terms of medicine have all that knowledge were found in zebrafish, because zebrafish is transparent for the first stages of life. There are some other modified lines that are totally transparent all their life. But yeah, basically all the embryogenesis, I don't have it here. But yeah, all the embryogenesis, we can determine it in even 3 days. you can see everything, what it's, all the organs, the development of the organs in the fish. And as we share many genes with zebrafish, many of them, the information that we know about humans was based on zebrafish.
Oh, okay. That's quite interesting. I would have never thought that
Yeah.
happened, but I've got another question. It might be a little bit or it's different from this last one. So, you told us that there were different parameters that affected the fish behavior, that gave some anxiety-like response. Did the hemp-derived compound affect anything else besides this? Besides this?
This is the only essay that I measure.
Okay.
So the thing that we, let's say we control the anxiety or not anxiety like behavior, it's with the distance. So the distance does not show us how anxiety or how anxious is the fish, but it gives us how, if it's affecting, for example, the the movement. In terms of benzodiazepines, they reduce your movement, reduce your low, and produce dizziness or produce somnolence. So in these terms, we measure the distance. So okay, yeah, it's for example in the diazepam, they show an anxiolytic effect, good, but they also show a sedative effect. So yeah, it's helping me in some way, but in other way, it's not helping me that much. In the terms of hemp oil or hemp extract, we have the opposite way from diazepines. They show an anxiolytic effect, but with an increment of distance move. So they cause hyperactivity, which it's also like, we don't want to be hyperactive for sure. So what we want is some molecule or some drug that reduce the anxiety without modifying the distance, or let's say it's not sedative or hyperactive, from two ways.
Okay, thank you. Now, as a kind of follow-up question, if I take it that you did some preliminary experiments, then you fed that to your machine learning or AI model, and then you keep going like that for quite some time. So, in your view, how could this machine learning model or AI model could reshape either early stages of research or screening or overall pharmacology in as you've been doing it?
Yeah, in like reduce the time of the process, let's say. It's just learning. Definitely we need something to feed the model to learn. That's why we download, and there's many databases. In this case, we have the molecules binding this receptor or this target. And if I cannot think of any other example right now, but I'm collaborating as well to a machine learning project that they are using the videos of the fish swimming in a maze. So I'm just recording videos, and they use these videos to feed the model. So the model can learn the behavior of the fish in terms of the molecules. And what I did, it's the you learn, okay, I have a value of 10. Its affinity, it's 10% affinity, let's say. So where does this 10% came from? Okay, so as we generate 200 descriptors of this same molecule. So it's gonna learn these 200 descriptors. How come those 200 descriptors came to this number, this affinity number? And same with the thousands of molecules. So that's the learning process. The model learns with the 1,000 molecules that you feed them. You test them in this 10-fold cross-validation. It's giving the model 90% of your data and testing with the other 10% how accurate it is. And then you rotate your data, and then you give another 90% of your data and you test it with the resting 10%. And then you do that rotation of data 10 times to see and to measure the error. And the model that showed the less error is the one that I took to make a prediction of my of the affinity of these molecules.
Okay, that's quite interesting, very interesting, I would say. That kind of gives some insight for some other researchers that might be doing the same thing or a very similar thing, or might need some tools for this. I am wondering how reproducible are the CRISPR-induced phenotypes across the different zebrafish generations.
We stabilize the line. So when we inject, you can inject and create a mutation. But every injection has a different mutation because it's the cast is, let's say, a pair of scissors that cut, and the cast you put it with a guide. So you tell them, okay, I want you to cut this part here. You're gonna cut. So the cast finds the where the guide is, the gene, and then the cast cuts. But it's that's like it's an enzyme, so you cannot predict exactly the enzyme where it's gonna cut. So once that you inject, all your fish has a different mutation. So you need to outcross them with a wild type animal, and then you have a half mutated and half normal animal or half wild type animal. In this stage, you extract a little bit of DNA and you clone it, you put it in a bacteria and send to sequence. And then the sequence is going to tell you, okay, this is your mutation, like punctual. Here, this nucleotide was cut, or this part of the gene, or maybe some nucleotides were inserted, or what was the modification. Once that you had this modification, there are predicting softwares that you put the sequence, and you can have the possible protein obtained from that mutation. So for example, I have one that was 19 deletion or insertion, I don't remember, but once I put it into the predictor, shows that has the same protein. So doesn't matter that has too many base pairs induced in the gene, but it still produces the protein. And my line, it's 10 base pairs deletion. So with this 10 base pairs deletion, it produces just half of the protein. So with this half of the protein, I know that it's not gonna function because it's truncated the half of the principal bindings of the protein. So yeah, once that you have them, you have your half wild type and half mutated. And if you find another one with the opposite sex with the same mutation, you cross them. If not, you just cross them again with wild type, and you have a bunch of half, and you now cross them using the Mendel laws of plus minus. So let's say or mutants are minus because do not work. The wild type are plus, and the heads, the heterozygous, has one allele plus and one allele minus. So I cross my heads, and I have 25% mutated, 25% wild type, and 75% half-half. So from them, I just have the ones that have the mutation, and I have all these mutated fish, and I just inbreed them. So I inbreed them to assure that all the babies are gonna have the mutation and all these other babies are gonna be normal.
Okay, thank you. Thank you very much. I would love to keep going on with questions, but I'm conscious of time. Dr. Del Rio, I don't know if you've got any other question, or perhaps in the YouTube transmit live. I'll leave the floor to you, Dr. Del Rio.
Thank you very much, Farid. Thank you very much, Jhosadara, for this really wonderful and deeply talk. It's amazing, all the things that you are doing. And thank you very much to you, and thank you to everyone joining us today. We need to have to go. And thank you for your time, your curiosity, and your questions. These sessions thrive because of the community that believes in science as a bridge, not just between countries, but between ideas, between persons. And we invite you to stay connected with us in all the forms. We are in the social media channel like UNAM UK, and MexSoc is also in Instagram, Facebook, X, and LinkedIn as MexSocUK, the Mexican Society of Students in UK. Thank you, and please be your engagement and this ecosystem to be brand intention and to share knowledge. We look forward to see you in the next session. Thank you very much.